Okay, so you guys can hang on to your exams. I'm not going to recollect them. The final is cumulative, and I like to pull a lot of questions from old exams. So it's a really good study guide for you. Other stuff, since we didn't meet last week, hopefully everyone saw the posted lecture or at least looked at the slides on mitosis, or the whole cell cycle plus mitosis. I want to do a quick review just so we at least have the stages of mitosis down. That's kind of the main, one of the main parts of that whole chapter. We'll look at some slides real quick. And as a reminder, your next exam is already next week. So the information we'll go over today is on cell membranes basically and how stuff moves across them. And then that will wrap up everything that will be on your next exam. So it'll be like cell structure, all the organelles, cell theory, and then the mitosis and cell cycle stuff from last week, and then this diffusion and cell membrane information. Okay. I have a few other things to mention kind of before we leave today, but I'll get to that, like upcoming other stuff. Okay. So we're actually I'm gonna review mitosis real quick and then just very briefly, we're actually gonna get the lab started because we need to take measurements at 30 minutes and 60 minutes on these potatoes, which I'll talk about in a little bit. So we'll do this first, then kind of group up and get started on that. All right, so mitosis, kind of the most well-known part of the cell cycle. Can anyone remember what the first stage of mitosis is? Start with that. We'll get the stages in order, and then I'll show you images of each of them in a cell. The phases, yeah.
Prophase.
Yeah, so prophase is going to be number 1. And that's what this cell is in. So this is a slide of a bunch of different animal cells. They're not all in the same stage of mitosis. So mitosis is just the dividing of one cell into 2 cells. They're all kind of in different stages of the cell cycle. This cell here, which the pointer is pointing at, is in prophase. The distinguishing characteristics really are that you still see a distinct nucleus, and then the chromosomes are kind of starting to condense. If you haven't watched the lecture from last week, this might make no sense at all. So definitely watch that, or at least look at the slides if you haven't yet. So that's going to be, this is prophase. I don't have the second one. Does anyone remember what the second?
Prometa.
Prometa, yeah. So I don't have an image of that, just because it's really hard to distinguish. I won't ask you to identify prometaphase, just know that it's in between prophase and metaphase. All right, so we've got prophase. This is my favorite stage. What's the next stage? Yeah, metaphase. So now this cell here is in metaphase. Metaphase is my favorite because it's the easiest to identify. The chromosomes are lining up along the midline of the cell. So you don't see a nucleus anymore. There's no membrane. They're all lining up and they're about to be split apart. So the lining up along the midline is metaphase. Okay. And the next stage, what's the name of that one? Anaphase, yeah. Anaphase, the chromosomes are now moving to either side of the cell. So you kind of see the cell elongating too, because it's about to elongate and then split into two cells. So you have the chromosomes here sliding to opposite ends of the cell. All right, and then what's the last one? Telophase. Yeah, telophase and cytokinesis. Those are often just grouped together because they kind of almost happen simultaneously. Telophase is technically the final stage in mitosis, but cytokinesis is where the cell actually splits in two. So now you start to see the cell kind of dividing in two. It's pinching in the middle, and then it'll eventually become opposite or separate cells. So the chromosomes have totally separated from each other. They're ready to divide. So we had, these are pretty easy to remember. Prophase, prometa's in the middle. And then metaphase. Anaphase and telophase. So you want to know that order and then be able to identify cells in all the stages except for prometaphase. I'm not going to ask you prometaphase, but I don't think I posted these slides yet. I'll post these slides to Canvas so you can look back through them. And at the bottom in the notes section, there's the answers. So you know, well, it's in order. So prometaphase, prophase, metaphase, anaphase, telophase. I always remember PMAT. I think I said this on the video. I never learned when I was learning mitosis, prometaphase wasn't a thing. So now it's PPMAT, I guess. It's an easy kind of way to remember the stages. All right. Any questions about that? I feel we were going over it considering I didn't really, I guess I talked to you about it from afar, but okay. That's a quick review of, quick review of mitosis. Now we're going to get the lab started. So this won't make a whole lot of sense yet, but we're going to so we're doing lab 6. I should say that. It starts on page 16 in your lab manual. We're going to look at how stuff diffuses and moves across membranes because that's what we're talking about in lecture today. So the part that takes a while is we have to let little chunks of potatoes sit in water with differing salt concentrations. The idea is that differing salt concentration is going to make water move either into or out of the potato for specific reasons that we'll get into. So we're missing one person. I was going to have 3 groups set up. So one person will just be on your own. You don't have to actually make the salt mixture because we're doing one is just going to be 0 grams of salt. I will say, I should mention this. This is usually done with something called dialysis tubing. That's what's in the lab manual. We're scrapping that and we're just looking at how salt moves into and out of a potato versus into and out of dialysis tubing. I think this makes a little bit more sense and it's a lot simpler to do. So the hypothesis 3 and 4, it'll talk about dialysis tubing. The concepts are the same. I would say just take notes on what we're doing on the potato stuff. Just ignore most of what's in there. We're doing this, it's the same idea, just a different structure that we're looking at water moving into and out of. So what I have set up over here, we've got, just ignore this for now. We'll talk about that in a little bit. This is going to be a beaker with no salt in it, just plain water. And this is going to be a beaker with 2 grams of salt mixed in. And then this will be a beaker with 4 grams of salt mixed in. So increasing salt concentrations, obviously. If I could get, let's see, 2 groups of 2 people. So if whoever wants to be on their own can be on their own. I don't really care. But then I need 2 people to make the salt mixtures and just weigh out 2 grams of salt and put it in here. And then 4 grams of salt and put it in here. So do I have any volunteers? Okay. I guess only, yeah. Okay, so yeah, we're on 6. Yeah, lab 6. So I have 2 scales, just as close to 2.0 as possible there. And then whoever's doing the 4 grams, you can dump it in the trash if it's too much. Yeah, it's not a whole lot of salt. And then 4 grams into here, there's only 1 salt container. I really only, oh, that's okay. Just take notes on what we're doing. I'll kind of lead you along. Okay, so you got 2, so yeah, put that in the 2 gram one and then mix it up with that little metal thing and just get it dissolved. I think you're good. You don't have to do any measuring. There we go. Wait, we will have to measure, sorry. We need to weigh the potatoes. That's okay. All right, so now, once we have the salts ready, and then this is gonna be our 0 gram, we're just plain water. There's 3 chunks of potato right there. Grab 1 for each of the beakers, weigh it before you drop it in. So get a like a 0-minute weight, basically. Write down that weight. And we'll share this data. You guys don't have to know all of it. Okay, and then, yep, drop it in there. Yep. And then we'll leave it. What time is it? 5:12. Sure, yeah, that's a good idea. Good idea. Okay, and then you guys weigh that one.
Weigh the potato.
Yeah, weigh the potato, not the, yeah. Yeah. Write that down and just remember what beaker you're at. That's going to be your station and you'll be responsible for weighing the potato chunk later. And I'm going to set my timer for 30 minutes and then drop it in. Yeah, drop it in the salt. You can take the stir out. The stirrer. Yeah. Okay, cool. So that's the start. Easy enough. Oh, sorry. I should have handed this out. Somewhere for you guys to write those. It's shovel today. We're not going to do the 15 or 45-minute measurements, so we'll cross those out. We'll just do 30 and 60. It'll give us enough info. Okay. Yeah, and then we'll get into some of the other parts of the lab once I start lecturing, just to take a little break. Okay, now we're going to get into cell membranes. All right, so the last part of the cell structure stuff that we're going to talk about is going to be the border around the cell that kind of keeps it all together and lets certain stuff in and certain stuff out, but not other stuff. Okay. So the plasma membrane, I'll probably usually call it the cell membrane or just the membrane. Technically, it's a plasma membrane. We generally talk about it as a fluid mosaic. So it's fluid in that it kind of moves a little bit. It's not completely static and solid. And the mosaic aspect, if you know art, a mosaic is just kind of a mix of a bunch of different tiles or different materials. And that's exactly what the plasma membrane is. So there's a bunch of different types of molecules that make up the plasma membrane. It's not just one. We'll focus on 2 of them in particular. the proteins and the phospholipids. But you can see here, it's just kind of a mix of different types of particles. Whoops. Okay, so fluid mosaic model is generally what biologists use to kind of wrap their head around what the plasma membrane is. So the major components we've got, like I said, oh, I'm sorry. We've got lipids and proteins. There's some carbohydrates in there, but I don't really care about them. We're not going to talk about them. So I want you guys to be familiar with the lipids that are in the plasma membrane and then the proteins. The two lipids are going to be phospholipids, which we talked about. Well, we talked about all of these in that biological macromolecules chapter. Phospholipids and cholesterol are the two big lipids. And for proteins, there's a number of different kinds of proteins, and I'll talk about the different functions of them very briefly. But we're really going to focus in on the proteins that help to transport stuff across the membrane. That's what most of this chapter is really about, moving stuff into and out of the cell. And that's what proteins help a lot with. So we'll talk about those proteins in particular. Okay. So first, I'm going to focus on the membrane lipids, the phospholipids, and the cholesterol. Most, if you counted up all of the molecules in a cell membrane around the entire border, the vast majority of them are lipids. So lipids make up most of a cell membrane, at least by count. So they're 98% of the plasma membrane molecules. Of those 98%, 75% of those are phospholipids. So phospholipids really make up the bulk of a plasma membrane. There's a few really important terms that are used to describe these phospholipids. So I'm going to go through them and kind of describe why they're important and why it matters. And this will come into play when we talk about what can move across the membrane and what can't. So their properties keep certain things out and let other things through. This is generally how a phospholipid is drawn. The head with two little tails. These are fatty acid chains. The tails are hydrophobic, meaning they don't want to be near water. They are going to repel water. The head, which is mostly the phosphate, is hydrophilic, which means it's fine if water is up against it. Having those two sides, a hydrophilic and a hydrophobic side, makes them what we call amphipathic. So amphi, like an amphibian, lives in water and on land. These also kind of have two sides where they have the hydrophilic and the hydrophobic. That's where the amphi comes from. Amphipathic molecules. And they're arranged in a bilayer. So I want to describe this bilayer pretty quickly. In this diagram, so here we have the cell membrane, a bilayer of phospholipids. This would be like the outside of the cell, outside of it, the extracellular area. And then this would be inside the cell. Both of those fluids are mostly water. So you have mostly water outside the cell and inside the cell. That means these hydrophobic tails that are scared of water are going to point towards each other, away from the water. The hydrophilic heads are going to be up against the extra and intracellular fluids. They're fine with touching water. The hydrophobic tails are not, so they point towards each other. You kind of have to have this bilayer. Otherwise, if you just had one layer of phospholipids, water would be repelled away constantly from the hydrophobic tails. So they create this sort of hydrophobic pillow in between. Okay, so I talked about hydrophilic, hydrophobic fluidity. These molecules will actually switch places with each other, which creates kind of that fluid mosaic. So they'll flip laterally, or they'll actually flip with each other vertically as well. So they'll just switch places with each other often. Not really going to go into why, but they do. They move around. And that's where cholesterol is going to come in. It'll have an effect on how much they move around. Let's talk about cholesterol briefly. So cholesterol makes up about 20% of the remaining phospholipids. Cholesterol is a lipid. I mentioned it, I think, when we went through the lipids before. Yeah. It's amphipathic. So it also has that hydrophilic head and hydrophobic tail. It just looks a little different. So you have all these phospholipids here, a typical structure. These little things stuck in between are the cholesterol molecules. So they have a tiny little head, but it's still hydrophilic. And then this big hydrophobic tail. So they're arranged kind of the same way, but they're interspersed sort of randomly in between the phospholipids. The whole point of cholesterol is to keep that cell membrane from being too fluid. You don't want cell membranes that are too fluid. Obviously, you don't want cell membranes that are too solid, so too much cholesterol, not a good thing. But too little cholesterol also causes issues. So cholesterol, as I mentioned here, the whole function is to reduce fluidity, keep it a little bit more stable. This is only in animal cells. Plants don't have cholesterol. So people who eat purely plant diets have very low cholesterol because they don't take any in from animal sources since plants don't have any. Okay. Those are the lipids. That's all I'm going to talk about with the two lipids, the phospholipids and cholesterol. On to the proteins. So membrane proteins. I'm going to start out giving kind of some background, then we'll get into the transport proteins specifically. So 98% of that plasma membrane, if you counted up the number of molecules, was lipids. The other 2% is proteins. However, so there's not many proteins, but they're really big. They make up about 50% of the weight of all the molecules in the membrane. So they're few and far between, but they're huge, and they have a really important role. We're going to talk specifically about proteins that span the entire width of the plasma membrane. Those are called transmembrane proteins. There's some that don't, but we're not going to worry about them. Okay. Fast. As I said, there's a lot of different things that proteins do. We talked about proteins in general, what their functions are. So some of these probably look familiar: enzymes, signal transduction. As I mentioned, the rest of this lecture is really going to be focused on transport proteins, proteins that move stuff into or out of the cell, and how they do that. I just want you to be familiar with these other 5 functions generally, just be able to list them essentially. But we're not going to go into any detail on how they do that. We just don't have time for it. But they do help chemical reactions happen faster. So they act as enzymes. They will send messages from outside the cell to inside the cell. They won't send this molecule in. That would be a transport. But a signaling molecule like a hormone would attach to one of these transduction proteins, and that would send an electrical message into the cell. So that's called signal transduction. The next 2 are kind of similar, a little bit different, but proteins will help cells recognize other adjacent cells. So kind of like a flag saying, oh, hey, I'm a liver cell or I'm a heart cell, and they recognize each other that way. They can also join in that same way. So kind of like fit together like 2 puzzle pieces. Those proteins will help 2 cells join together so they stay a little bit more solid. which is important in some tissues. And then finally, proteins will attach to this skeleton outside of the cell, the cytoskeleton. It'll basically just help anchor the cell. So cell anchoring, cell joining, recognition, sending signals into the cell, and then helping chemical reactions happen, along with the really important one, transport. That's what we'll talk about. Membrane transport. We're going to focus on here. You can go back and look at those comics if you want. Okay, a little bit more terminology before we get into how this actually works. Plasma membranes are referred to as selectively permeable. Permeable in that they let things in and out, but they're selective about it. They don't want anything to come in or anything to go out. They want to be able to control it. So they're selectively permeable. That's the term that's used. And they can move things across them in one of two ways, either using energy or passively not using energy. So passive transport is the kind that does not require any energy. And I know I mentioned ATP before, but just a reminder, ATP is the cell's energy. So when you see ATP, think energy. There will be ATP like lightning bolts on some of these slides when energy is needed. So get familiar with that, and that'll come up a lot in the next unit as well. So passive transport, no energy required, which is good for the cell. It just happens due to the random molecular motion of particles, which doesn't probably make a whole lot of sense right now, but we'll talk about how generally how particles tend to move on their own if they're not forced to go one way or another. There are a few different types of passive transport. One is diffusion. You can have simple or facilitated. I'll talk about what each of those means. And then osmosis is specifically the movement of water, which doesn't require energy. It's always passive. But osmosis is water moving in and out of the cell. And that's what we're looking at in the potato lab, actually, how water is going to move into or out of that potato. Active transport, there's really only one kind. I'll talk about some detail on it, but it consumes ATP. So the cell needs to use energy to move certain things across the membrane. And I'll talk about when that happens. All right, let me check on something real quick. Very simple. Before I get into what's happening here. So what I did over here, this, let me stop sharing. So this beaker that is on a hot plate, so that's hot water. I put something called potassium permanganate in there. It's just a crystal that has a pretty color, basically. And you can see how it's dissolving, and you can actually see the little like clouds of potassium permanganate coming up. That's obviously room temperature water, and I put the same thing in there. You can see it spreading out a little bit. Feel free to walk up here if you want to. But you can see in the hot water that those those potassium permanganate crystals are dissolving and then doing what we call diffusion, which I'm about to get into, but they diffuse throughout the water. It happens a lot faster when the water's hot. So if you think about, and this happens with any molecule, so you can think about it with like water vapor or oxygen in the air, whatever you want to think of. Molecules of whatever substance you're talking about, in this case, potassium permanganate, are going to want to spread out from each other. So they want to go towards where there's fewer of them. It happens a lot faster when they have some heat, just because it increases their energy. So hot water is going to increase the rate of diffusion. This is the random motion of molecules. So that's just what they do. I didn't put anything that added energy into there. I just let them do what they were going to do, right? So this would be passive. This would be a passive form of, well, motion. We're not putting anything across a membrane right now. But heat is going to speed that process up. So that's an important part of this aspect of the lab. And you can watch this over time. That's pretty much already made that really hot. It's already completely diffused, and then we'll see kind of what happens in the cooler water over time. But we can come back to this and revisit it. But that's the idea behind diffusion. Those molecules are just bouncing off of each other and trying to kind of get away from each other. That's essentially what molecules do if left up to their own devices, which is what I'm about to talk about. Any questions about that? All right, back to lecture material. We're going to spend a lot of time talking about passive transport here, because there's obviously multiple kinds. So if you imagine this passive transport, this is kind of a diagram of what we'll be talking about. Passive, you have a couple different kinds, simple and facilitated. And then active transport, you see that ATP lightning bolt. That means energy is needed to make this one happen. We'll get into that in a little bit. In the first part that I'm going to go over, passive transport, specifically simple diffusion, which is what's happening there with that potassium permanganate. Molecules are just moving on their own through a cell membrane. They don't need any help whatsoever. They can just skirt right through those phospholipids. In facilitated diffusion, don't worry about the two different shapes of the protein here. Facilitated diffusion, a protein is needed. So no energy is needed, but you need a protein to kind of create a channel for these molecules. So they need some help getting through. They can't make it through the bilayer on their own. And then, yeah, active transport, you need energy. So we'll get into that. This looks like a confusing diagram, and maybe it is. I'm going to go over the important parts. This is basically showing what will move through a membrane through simple diffusion. Stuff that doesn't need a protein to help it. And it's really just from here up. Everything below there is going to bounce right back off, which you can see those arrows. These guys can't make it through. The most important aspect, the most important part here, hydrophobic, nonpolar molecules, so same thing. Hydrophobic molecules are nonpolar. They repel water. They don't mix with water. They can go through just fine. They can be big, small, anything in between, and they can make it through the membrane. They don't need a protein. They don't need any help getting into the cell. Water can kind of move through, but also bounces off. We'll talk about a protein that helps water a little bit. Any idea why water wouldn't be able to make it through, whereas nonpolar molecules can? What is water? Polar. Yeah, water is polar. Nonpolar, this whole area in here is nonpolar. They're hydrophobic, those tails. So anything that's down here, polar, which water is going to have a really hard time getting through because it's kind of bouncing. It's trying to kick anything that's polar back out, essentially. Water's small enough that it can kind of eke through a little bit. All of these other things, anything that's large and polar, like glucose, no way, not making it through. It needs some kind of help getting through, so it'll bounce right back off. Anything that's charged, these ions, also very repelled by this hydrophobic section. And then obviously anything that's charged and polar is not going to make it through. Non-polar makes it through, polar doesn't make it through for our purposes. Okay. Into more detail about how this works. I'm going to talk about simple diffusion, those things that can make it through on their own. So anything, we'll imagine anything that's non-polar can just make it right through. We're going to talk about how that happens. These are 3 of the important bullet points, and at least one will be different from the other types of transport. So this is kind of a good way to figure out, okay, I need these 3 things to be true in order to have simple diffusion happen. If one of these is not, it will be one of the other types of transport. So no cellular energy is required. We talked about that. We know we're in the passive transport still. I'm going to skip down to the 3rd bullet point here. No transport protein is required. We just talked about that. These are nonpolar molecules. They don't need a happy channel to get through. They don't need a tunnel to go through that phospholipid bilayer. They can just go right through without any issue. And one of the important parts here that I haven't mentioned yet is how molecules move. I kind of referred to it with that potassium permanganate. The term for molecules moving from an area of really high concentration where there's a lot of them to low concentration, like spreading out, is called moving down the concentration gradient. And you'll see that term and I'll ask about it and ask you to kind of interpret it in different ways. So molecules, just like that potassium permanganate, are eventually, over time, even in colder water, they're going to go from where there's a lot of them to where there's not a lot of them. And that oftentimes means going across the cell membrane. So as long as there's a high concentration out here and a low concentration in here, those molecules will just make their way in naturally. That's why no energy is required. That's just what they do on their own. I didn't put any energy, well, I put energy in by heat to the hot version, but in the cold version of that water, I didn't put any energy in, and the molecules are still dispersing. I think of them as being like antisocial. They just don't want to be around other molecules. They want space. They want to get out of the rat race. So that's just how molecules move, that random motion of molecules that I mentioned earlier. When a cell wants to do the opposite and basically kick something out against the concentration gradient, that's when you need energy. That's when you get into active transport. So we'll get into that. But if molecules are moving down their concentration gradient from high to low concentration, that's going to be passive transport. In this case, simple diffusion, because we don't need a transport protein. Okay. The next topic, if you understood that, this one's pretty easy. Facilitated diffusion, same thing's happening, but now it's one of those groups of molecules that just can't make it through the membrane on their own. They need some help. So this is going to be facilitated diffusion. A protein will facilitate the diffusion and help it out. So still no energy required because we're doing passive transport. We know that because molecules are moving down their concentration gradient. So still that same general movement from high to low concentrations. And this is sort of a an edited view of that diagram that I had up earlier. So this is the lower half, all that stuff that basically bounced back off without the protein. Large uncharged polar molecules, well, anything polar, and then ions. They can't go through on their own. They need the help of these proteins, these weird shapes here. But they're still moving from high to low. That has not changed because we're in passive transport. A transport protein is required because, like I said, they just can't get through that hydrophobic area. They get bounced back off. They don't mix well. So actually, you don't need to know this, but in the middle of these proteins, there's a hydrophobic chain of amino acids, or sorry, hydrophilic. A hydrophilic group of amino acids. So it creates this like hydrophilic channel. So anything that is hydrophilic, which is anything that's not hydrophobic, can just scoot right through. Like a little, a tunnel through a mountain, or under an underwater tunnel, like the channel or something like that. You can imagine it. Normally things wouldn't be able to get from one side to the other, but these proteins are allowing for that. So that's facilitated diffusion. Still diffusion. It's still molecules moving from high to low. That's easy to forget. Any questions so far? I feel like I'm going quickly. Are you guys doing okay? Okay. All right. Finally, osmosis. This is our last kind of passive transport. As I referred to earlier, it can be simple. So not every water molecule needs a protein to get through, but it's a lot more efficient when you do have a protein. So it really helps a lot more water move into or out of a cell if there's a special kind of protein that I'll talk about in just a minute. So it can be simple or facilitated. And remember, osmosis is just water. I think I said that. We're talking about water movement here. With the other kinds, we were talking about not water. Okay. Osmosis is definition, the net flow of water through a selectively permeable membrane. So our selectively permeable membrane is our cell membrane in this case. You can set up selectively permeable membranes in experiments as well, which is sort of what the original version of this lab without the potato does. But water is, this is a little bit weird. It's a weird way to think about it. I think about it a different way. So I'll tell you kind of whatever makes the most sense to you for how water is going to move. We want to figure out, our goal with osmosis is to figure out essentially if water is going to move into a cell or if it's going to move out of a cell. And that's all based on the solute concentration. And by solute, I mean anything dissolved in there, like salt. That's why we put salt in that water. So I think of it as the second bullet point here. The solute particles are going to pull water towards them. Water goes towards the higher solute concentration. So where there's more salt, that's where water is going to move. Oh, it's about time to weigh our potatoes. So here you can see in this YouTube, a lot of salt on this side, that's the little triangles, and then the liquid is water. The water is going to move towards where there's more solute. So after a bit of time, you get more water on the left side. Only water can move across this membrane. The solute can't. And in any problem I give you or any question, I'll tell you what can move across the membrane. Sometimes the solute can, sometimes it's just the water. In this case, just the water can move back and forth. The solute has to stay on one side or the other. So that water is going to move towards where there's more solute. But essentially it's still diffusion. It's diffusion of water particles. Water is moving from where there's a lot of it to where there's less of it. If you think of water as individual molecules trying to spread out from each other. On this side of the tube, there's a lot more water. than on this side, even though the levels are the same. More space is being taken up over here by the solute. So there's actually more water over here, more individual water molecules. So that's also, it's another way to think about it. Water is going to go towards where there's less water. That doesn't really make sense in my head, but if it makes sense in your head, and that helps you figure out which way water is going to move, awesome. Whichever of these two makes the most sense to you, go with that. Okay, let's take a break here. Oh, get some paper towels. Everyone's going to weigh their potatoes. Kind of dab them off with the paper towel. They don't have to be super dry. Just get the water generally off of them. And right now will be the 30-minute wait. Only have 2 potatoes. Make sure you're writing down the wheat for the right. And then after you're done, just plop it back in the water and we'll take a measurement in 60 minutes. So hopefully you got some kind of change. Did everyone get a change? Awesome. Okay. So osmosis. Hopefully just from that, you can tell that water is either moving into your potato or it's moving out of your potato. And that is where the solute concentration comes in. We'll talk about that in more detail. Those special proteins that I talked about that help water move through are conveniently called aquaporins. They're like little pores for water. They are specific proteins that only let water in and out of the cell. So like I said, water can get through, but not very well without these aquaporins. But most cell membranes have a lot of aquaporins to help make it a little bit more efficient to get water into and out of a cell, which has to happen quite frequently. All right. Okay, this is where we get into something called, well, it's called tonicity. We're going to talk about solute concentrations. And when I say solute, just remember, I mean, you can imagine salt. So salt is the most commonly used example of a solute, something that dissolves in something else. We're going to be talking about the concentration of salt on one side of a cell membrane versus the other side, and what that does to water movement in or out. That's what tonicity is all about. You can also compare solutions to each other. So some solutions have more solute, which would be like one side of the cell membrane. And other solutions, which you can imagine is the inside of a cell, might have a different solute concentration. And cells want to keep a certain balance. Sometimes they want more salt inside, sometimes they want more salt outside, just to control, it's how they control the water movement into and out of themselves. All right, so tonicity is a general term for that. We have 3 options for what a solution can be, or in comparison to another one. So this is, oh, you guys can't see that. A hypotonic solution, so you have hypo, hyper, and iso. I'll go through them one by one. A hypotonic solution is a solution that has a lower solute concentration than the other solution you're talking about. So if we have A and B, if A has a lower solute concentration than B, it's hypotonic compared to B. It's always in comparison to something else. When you hear hypo, you can think low. Hypo, low. It's a low concentration. Hyper is the opposite. If someone is hyperactive, they have a lot of energy, right? They're very active. So a hypertonic solution has a lot of solute. So one solution, if you have a hypotonic solution, the other one automatically has to be the hypertonic solution. So you're always comparing two. Okay, so we have hypo, hyper, and then the only other option is that they have the same solute concentration. Oh, I didn't even put that on here. Okay, never mind. You don't need to know isotonic. We're not going to talk about isotonic solutions. So the movement of water, that's what we're trying to get at. We are going to try to figure out whether water is moving into or out of a cell. It's going to move towards the hypertonic solution. Like I mentioned a couple slides ago, water is going to move towards where there's more solute. So if you have a hypo and a hypertonic solution, it's going to go towards the hypertonic, towards the greater solute concentration. In our case, that, and for our purposes, that's always going to happen. Always going to move in that direction with passive transport. Hypo to hyper. That's where water is moving. Remember, we're talking about water movement, not the solute. It's really easy to get mixed up when you're thinking about stuff moving across a membrane. And I have a worksheet here for you guys to work on. In some of these, the solute is going to be moving across the membrane. So it's not always asking about water. Okay, here comes ISO. So sorry, this is a little blurry. This is just a simple diagrammatic representation of what I was talking about. Pictures always help me. I think I'm very visual. So here we have a cell, just like part of a cell here. These little green particles are going to be the solute. There's a lot inside the cell, and there's not a lot outside. So that cell is in a hypotonic solution. That's what we would say. So we're thinking of this as like a red blood cell here. If this is a situation in a red blood cell, water is going to move in, right? Water is moving towards that higher solute concentration. And that can eventually make a cell explode if there's too much salt in there and it draws too much water in. Isotonic, I didn't define, but iso means the same. So same amount of salt inside and out, kind of where animal cells want to be. So you have a nice balance of water inside and outside the cell. A hypertonic solution, which we actually created with those two salt solutions back there. So we put a lot of salt in the solution around the potato. The potato has less salt in it. Water will leave that cell to go towards the higher solute concentration. And that oftentimes makes cells shrivel up. Questions about that? Okay. If you're getting that, that's awesome. That's probably, I think, one of the more complicated concepts when we're talking about these cell membrane, the transport across cell membranes. Okay. So I kind of talked about animal cells. You don't have to worry about the term lysed, but if water is moving in, it'll explode. Animal cells, like I said, generally want to be in an isotonic setup. Plant cells are a little bit weird, so I wanted to take a little bit of time to talk about them really quick. So down here I have, yeah, the environment. So the environment, I mean around the cell. So what kind of solution is the cell sitting in? Hypotonic is what plants actually prefer. So if they, rather than an isotonic solution. So they actually want water to be moving into their cells to keep them rigid. So they need more water in their cells just to keep them upright. Because again, they don't have skeletons or anything to hold them up. So the pressure of that water in their cells helps them hold up. So the normal state where plants want to be, they want their cells in a hypotonic solution. And that is pretty much the opposite of what we're creating back there, right? With the salt concentrations at least. They kind of go limp if they're in an isotonic solution. And then if they're in a hypertonic solution, which we did with the 2 salt concentrations back there, they do something called plasmolysis, and I have a video to show you. Usually in lab, we look at it under the microscope, but I think you can see it a lot better. This is, I think, the very last part of that lab. Yeah. Page 18, hypothesis 5 is about plasmolysis, really about just what's happening with plasmolysis. I'll kind of describe why this happens in plant cells and not animal cells. So animal cells will generally shrivel up. All they have is a cell membrane. Plant cells have this nice rigid cell wall. And inside of that, they have a cell membrane. So they kind of have 2 layers. What's happening when they undergo plasmolysis is that cell membrane, it's shriveling just like this cell, right? So it's shrinking in, but the cells still stay rigid because of that cell wall. So that specifically happens in plant cells when they're in a hypertonic solution, plasmolysis. So this video doesn't have any sound. I'll try to kind of pause it and talk about what's happening. Right. Okay. So it's a little bit blurry, but these are plant cells. They're nice and rectangular. They look a lot nicer than animal cells, and they're all like packed up against each other. Okay, so here we're going to add a sucrose solution, and sucrose is just another solute. It's like salt. It's a sugar. We talked about it before, but if you add a sucrose solution, you're adding it to the outside of the cell here, right? So what do you guys think is going to happen? Okay. Yeah, well, it'll make the overall cell heavier. What's going to happen to the water? Is it going to move into the cell or out of the cell? What's that? If you put a bunch of solute outside of the cell, where's that water going to move?
The cell, it's the water ton cell will come out of it.
Yeah, the water's going to move out of the cell. It's moving towards this higher solute concentration. So you can kind of see, so you still see the rigid cell wall, right? So they're still holding up just fine, but this green part is kind of all the stuff in the cell, and it's starting to shrink in because the water is leaving. It shrinks down, and this is plasmolysis. So water is exiting the cell. It's not really all that impressive, I get it, but this is the process of plasmolysis. The cell wall stays rigid, but the cells overall, the inside, shrinks down. This is when a plant would like wilt, for example. There's not enough water in the cells. So you can see it shrinking down, and then it's really, everything's getting concentrated into the middle. So now they're going to add just pure water. So if we're going to add pure water, where's the water going to go? Into or out of the cell? Into the cell, right? Because now there is more salt or sucrose, I guess, inside the cell. There's not enough water. The water is going to go into the cell, and it'll expand back out. pretty quickly. So then if you've ever seen like a wilted plant and you put water on it, it kind of just re-ups. Okay. So that's plasmolysis. That's the last part of that lab. As long as you understand that plant cells will shrink down in a hypertonic solution because water is going to leave them, that's the whole idea behind plasmolysis. Okay. You guys want to take a break? Let's take a break. Okay. Next. Yeah, let's take like a 5-minute break and then we'll do a problem here. All right, are y'all ready to get started again? I don't know what time I said we'd have 5 minutes, but everyone's back. Okay. So this kind of problem is a kind of problem that I would ask you on an exam. We're specifically trying to figure out which way water is going to move. So I kind of like thinking of, if we're thinking of a cell, it's like a little balloon with a semi-permeable membrane in water. That's how you can kind of think of a cell in relation to hyper and hypotonic solutions. So here we're going to try to figure out, so the membrane around the cell is only permeable to water. So only water can go in or out. The solute that's in the cell has to stay in the cell. The solute that's in the environment, which I have listed here, in the water around it, has to stay outside the cell. So that doesn't change. It's just water. Is water going to go in or out? So does anyone want to venture a guess as to whether water goes in or out? You have a 50-50 shot. This is kind of complicated because I have multiple solutes, but yeah, exactly. Awesome. It doesn't matter that they're different kind of solutes. That doesn't really affect how water moves for our purposes. You have sucrose and glucose, and then you have sucrose, glucose, fructose. Who cares? It doesn't really, you can ignore all of the names. You just need to add up the concentration. So you may not be familiar with this unit, but just think of it as how much solute there is. In here we have 0.03 and 0.02. So that's 0.05, right? If you add that up, we have 0.05 solute in here. In the environment, we only have 0.03. So water moves into the cell because it's moving towards that higher solute concentration. It wants to dissolve that solute really bad. Does that make the environment hypo or hypertonic compared to the cell? Like before there's any water movement, just yeah, exactly. So hypotonic because it's a lower solute concentration outside the cell. Hypo means low. There is less solute outside the cell in the environment than inside. Awesome. So that would make the cell what? Hypo or hyper? Hypertonic, yeah. It's always got to be the opposite, right? If you're talking about two solutions, one's got to be hypo and one's got to be hyper. I'm not going to ask you guys about isotonic solutions. So it's one and the other. Okay, awesome. I handed out this diffusion and osmosis worksheet. I think this will help you guys kind of gain a little bit better understanding This is not, actually, it's mostly diffusion. I don't think I have anything about osmosis on here. It's all diffusion. So this is talking about solute movement, and that's not what we were talking about here. This is actually a little bit simpler than osmosis. So, I mean, we have plenty of time. Do you guys want to work on this? At least like the movement of the molecules right now before we move on. We're almost done with this lecture, and the lab will end pretty quickly as well. So I'd say let's at least get through to where, like, get through number 1 and 2, figure out where those solutes are moving. So this is where it's important to realize what the heck is moving across what. And we'll go over those answers. All right, so I'm gonna have you guys give me measurements. I have a graph all set up, so it should populate automatically, and we can kind of see what happened with these different salt concentrations. We'll see if this works. I tried it on Friday, and it worked with my fake data. Okay. So 0 grams, whoever measured like the pure water, what was the starting weight? 0.7. And then your 30-minute weight? And then 60. Cool, okay. Oh, there, sorry. You guys can write those in. So yours gained weight. It took water in. Would you guys have expected the potato to take on the water when it's just in pure water? Yeah? Why? Yeah, there's a lot of solutes inside a potato, like lots of different ions and stuff. There's none really in that water, like relatively. So all that salt inside the potato is pulling that pure water in, basically. So it took on water. Okay. Hopefully the next two are different. For the 2 gram group, what was your 0-minute weight?
21.
30?
20.5.
Sweet.
19.8.
19.8. Awesome. Yay. So just the opposite, right? Just 2 grams of salt is basically a higher salt concentration than what's in that potato. So now water is being pulled out of the potato by that salt in the water. Okay, let's see what the 4 gram did. What was the 0-minute weight on the 4 grams? 21.3. 30 minutes? 8. 60. 19.3. Good, awesome. Man, that works so well. It's so much more interesting than dialysis tubing. I know, like a potato, yay. It's not super exciting, but if you guys did the dialysis tubing, you'd see how boring it really is. At least this is a real thing. I mean, dialysis tubing is, but this is a real thing in nature. Okay, so we know why the potato in pure water gained weight, right? And we know why the potatoes in salt water lost weight. The important difference between the 2 potatoes and the different concentrations, obviously more salt in the water is going to pull that water out faster, right? So if we kept it going, these would kind of stay parallel until I guess all of the water would be pulled out until they hit a balance. I don't know how long that would be. I think I did this for 2 hours on Friday, and it at home. I wasn't here, and it was still separate, so it hadn't actually evened out. So the important part here is that the more hypertonic a solution is outside of a cell, the faster that change is going to be. So it's a steeper concentration gradient is what we say. Write that down. Okay. So this gray line is the 4 gram one. The difference between the salt concentration inside and outside that potato is a lot greater in that 4 gram treatment compared to the 2 gram. So we say that's a steeper concentration gradient, which means it's gonna pull even more water out. So it just has more of an effect. Probably not a huge surprise, but that is generally what we are talking about if you can read my writing. Steeper concentration gradient is going to mean faster loss of water, essentially. So steeper concentration gradient, lose water faster. Cool, that worked really well. I will post that completed data sheet. I need to make a note of all the stuff I need to post. I'll post that completed data sheet to Canvas as well. I guess to the lab section. I don't really post a whole lot to that, so you guys can see it and look at it as well. Okay. Mitosis slides. Awesome. Does that make sense? Did you guys notice a difference in the texture of the potato, which was like soft and flimsy? Yeah, because it lost a lot more water, right? It's kind of just like losing its life force, I guess, is a way to think of it. Yeah, it gets a lot flimsier. And then the pure water one is actually still pretty solid. Okay. Any questions about that experiment? So that covers hypothesis 3 and 4. 3 was generally which way is water moving, into or out of the potato in our case. And then the steepness of the concentration gradient was hypothesis 4. The more hypertonic the solution, the faster that water is going to be moving out of the cells. You're ready.
So we turn it in on this, like on this little lab up there?
No, you don't have to turn it in. Just for the quiz. Yeah. Yeah, exactly. Yeah, and on that note, next week we're going to do a review, and then the week after that is your first lab practical, which is just kind of questions covering the first 3 labs. I don't think this one is included in that. I kind of moved some labs around, so we'll basically go over all the important points from the first 3 labs next week, and you'll take an exam on it the following week. And I'll talk more about that, but it'll just be questions in here, probably slideshow questions. Okay. Yeah. Okay. Last little segment of transport. You're very close. Active transport. There's only one kind of active transport for our purposes. The defining characteristic is that it uses energy. All right. So now we are moving against or up the concentration gradient. I should have a slash there and say up. Both of those terms are used. Before we said down the concentration gradient. You could say with it if you want to. This would be against or up the concentration gradient. So now cells are trying to move molecules in a direction that those molecules do not naturally want to go, from a low concentration to a high concentration. It requires energy, ATP for our purposes. I already know that. I'm going to talk about one specific example, which is basically how our entire bodies work, the sodium-potassium pump, which sounds really uninteresting. And when you get into the details of it, it kind of is, but I'll talk about why it's so important. There's other examples. I just want you guys to know the sodium-potassium pump, so that's all I'm going to go over. Don't think this is the only kind of active transport there is. Sodium potassium pump. So we're going to look at it, an example to kind of see how this works. All right, there's a lot of details here. I'm going to try to walk you through them. And at the end, I'll say, okay, this is the main take-home message. I forgot to go through these slides and kind of cut some stuff out. But it's a simple concept, I guess. And I'll tell you what I want you to know at the end, like the main take-home messages. Okay, so here we are looking at a specific protein that's going to move sodium to the outside of the cell and potassium inside. So here you can see on this little protein, there's like 3 slots for sodium ions. On the other side, there's this other shape. That's where the potassium ions are going to go. So it's one protein that does both. In order for it to do this, it needs energy. The sodium concentration inside the cell, we'll imagine this lower side is the inside of the cell, is low. Outside, there's a ton of sodium. But we still want more sodium out of the cell. So that's why it's going to be active. So you need some ATP in there. So the sodium will basically get loaded up into the protein. Some energy will be added, and then those 3 sodium will be kicked out of the cell. So now it's an even lower concentration of sodium in the cell and even higher outside the cell. For each cycle, each time this happens, 3 sodium are leaving. I know it's very specific, but the difference in the number of sodium versus potassium coming in and leaving is important. So these are 3 positively charged ions that are leaving the cell. Okay, so we've got sodium leaving the cell. So here we're at number 3. That's what I showed you on the previous slide. Now we're kind of going in this cycle here. So step 4, first off, the potassium, which is K, potassium concentration in the cell is high. Outside the cell is low. We want to make it even higher in the cell and even lower outside the cell. So each cycle will bring in 2 potassium ions. So you see them here on that protein. So for every 3 sodium that go out, 2 potassium come in. Both are moving against their concentration gradients. They're both moving to an area of high concentration. So then, yeah, you have the potassium loaded onto the protein, and then they get spit out inside the cell. It's a fairly simple concept. 3 sodiums out, 2 potassium in. And then if we look at it all together, so putting all of those steps together, so we'll start with 1 and then make our way around here to 6. if you want to see it all at once. So this is the sodium potassium pump. We're kicking 3 sodiums out. You need ATP for that. Potassiums will come in using kind of another form of energy. So we get 2 potassiums in. 3 sodiums out, 2 potassium in. That's a really important concept that I want you guys to remember. They're both positively charged ions, which I think on the next slide, I'm going to talk about the importance of that. So you're basically losing more positives than you're gaining. So you're changing the charge inside that cell. Oh yeah, 3 sodium out, 2 potassium in. Okay, great. Okay, so why do we care that 3 sodium are leaving the cell and 2 potassium are coming in? So I basically just answered this question. If you're losing 3 positive ions and you're only gaining 2, what does that do to the charge inside the cell? Yeah, it brings it down. It makes it negative, right? You're losing more positives than you're gaining. So the charge, if you think all of our body runs on electricity, basically. So this charge inside the cell is really important. So these cells want to have a negative charge inside. All of our cells want, most of them, want to be negatively charged inside. It's really important for their functioning. I'll talk about why in just a second. This is called a charge differential or an electrochemical gradient. So we have concentration gradients where there's a higher concentration on one side of the cell versus the other. Electrochemical is just as it sounds, a chemical, so an ion in this case that also has an electric charge with it. So cells are building up an electrochemical gradient within them. So again, why do we care? I haven't quite gotten there. So this voltage across the cell membrane, it's weird to think about our cells and all living cells as having this voltage, but we have a voltage in our cells and it creates potential energy for future release when needed. So what do I mean by that? If you imagine a cell and it's getting more and more negative inside, you can think of it like a battery. So you have a negative side and a positive side, right? We know that can create stuff. It can create energy, it can create light, it can do a lot of different things. So with our cells having that negative inside and positive outside, it's the same idea. It creates a potential source of energy. They can start to release that negative. And then that's essentially what allows all of our muscles to contract, our heart to beat, our nerves to fire. Most of what's going on in our body is because we have inside our cells a negative charge, outside our cells a more positive charge. The whole point of the sodium potassium pump is to build up that negative charge inside our cells. We need a negative charge inside our cells so we eventually have energy to contract our muscles or think or feel something that pokes us, etc. It's a little bit strange to think about, but we just run on this electricity, this difference in positive and negative charges in our tissues. So that's why the sodium potassium pump matters. It keeps us going. If we had the same charge inside our cell as outside, we'd be dead. We wouldn't be able to do anything. Our muscles wouldn't contract, which means our heart wouldn't be pumping. All of the other muscles that need to contract for bodily functions wouldn't do that without this charge differential. The sodium potassium pump is in almost every single cell of our bodies. There's more in certain cells that need to contract more. Yeah. That's why moving 3 sodiums out and 2 potassiums in matters. Hopefully you buy into that. Questions? All right, I got rid of a few slides. That's it. Okay, so that wraps up all the material for the exam next week. Same setup for the exam next week. It'll be 40 questions, 35 multiple choice, 5 fill in the blank with a word bank. I have a Kahoot review quiz. Do you guys want that posted again? Okay. I will post that. Put that down, and then I'll post the assignment for the diffusion worksheet so you guys can take a picture of that and upload it. The Excel spreadsheet for the lab we just did, and then the mitosis slides.
The exam gonna have a lot of pictures on it.
Yeah, so there will be like diagrams, kind of like what's on that diffusion worksheet. Mitosis, yeah, there will be mitosis pictures on there. Yeah, there will be slides of mitosis. They'll be as obvious as I can find them. I don't like to make them confusing. So if you know what happens in each stage, and you kind of know what it looks like, you should be fine. It won't be like an in-between.
I haven't looked. Is there a study guide over there?
Oh, I don't think I put up a study guide now. Thank you for the reminder. Yep, I'll put that study guide up as well. Probably tomorrow. Let's see. What else? What else? So you got the two. Okay. Do you guys want to go over anything with that lab? You think you got it more or less? I know you were saying the lab quizzes are kind of throwing you off. Any questions about the lab? Okay. I feel like I'm forgetting something. Yeah, so no lab next week. We'll just do a review for the practical. Yeah, I've probably forgotten something major, so I'll email you if I did. But I think that's it. Everyone has their exams, right? If you have questions about any of those questions, just let me know and I can help you out.
You said the finals cumulative, so what would it be?
The finals cumulative, so it's over everything from the first day of class, lecture, not lab. There's nothing cumulative in lab, except like the lab practical that you're gonna take in a couple weeks will be over the first 3 labs we did. So yeah, I like to pull questions from old tests. Yeah. I think that's it. Any questions? Okay, I'll see y'all next week. Yeah, we're good.